Sudan virus is one of 5 known viruses within the genus Ebola and causes Ebola disease in humans and other primates. It is the sole member of the species Sudan. Ebola/SUDV is a select agent. World Health Organization Risk Group for Pathogen. National Institutes of Health, National Institute of Allergy and Infectious Diseases Category A Priority Pathogen. Centers for Disease Control and Prevention Category A Bioterrorism Agent, and listed as a biological agent for export control by the Australia Group. The first known outbreak of EVD occurred due to Sudan virus in South Sudan between June and November 1976, infecting 284 people and killing 151, with the first identifiable case on the 27th of June 1976. Use of term Sudan virus was first described in 1977. It is the single member of the species Sudan Ebola which is included into the genus Ebola family Filaviridae, or order Mononegavirales. The name Sudan virus is derived from South Sudan and the taxonomic suffix virus. Sudan virus is pronounced Sdane v Reza Sudan Vahi Ruhs in English phonetic notation, according to the rules for taxon naming established by the International Committee on Taxonomy of Viruses. The name Sudan virus is always to be capitalized, but is never italicized, and may be abbreviated. Previous designations. Sudan virus was first introduced as a new strain of Ebola virus in 1977. Sudan virus was described as Ebola hemorrhagic fever in a 1978 WHO report describing the 1976 Sudan outbreak. In 2000, it received the designation Sudan Ebola virus, and in 2002 the name was changed to Sudan Ebola virus. Previous abbreviations for the virus were EBOVS and most recently SEBOV. The virus received its final designation in 2010, when it was renamed Sudan virus. Virus inclusion criteria. A virus of the species Sudan Ebola virus is a Sudan virus if it has the properties of Sudan Ebola viruses and if its genome diverges from that of the prototype Sudan virus. Sudan virus variant Boniface by 10% at the nucleotide level. Disease. SUDV is one of 4 Ebola viruses that causes Ebola disease in humans. EVD due to SUDV infection cannot be differentiated from EVD caused by other Ebola viruses by clinical observation alone, which is why the clinical presentation and pathology of infections by all Ebola viruses is presented together on a separate page. In the past, SUDV has caused the following EVD outbreaks. Ecology. The ecology of SUDV is currently unclear and no reservoir host has yet been identified. Therefore, it remains unclear how SUDV was repeatedly introduced into human populations. Bats are suspected to harbor the virus because infectious Marburg virus, a distantly related filavirus, has been isolated from bats, and because traces of the more closely related Ebola virus were found in bats as well. Molecular biology. SUDV is basically uncharacterized on a molecular level. However, its genomic sequence, and with it their genomic organization and the conservation of individual opened reading frames, is similar to that of the other 4 known Ebola viruses. It is therefore currently assumed that the knowledge obtained for EBOV can be extrapolated to SUDV and that all SUDV proteins behave analogous to those of EBOV.